So it is my pleasure to reintroduce to the group Dr. Margot Taylor, who is a senior scientist at SickKids, and she's the head of the functional neuroimaging lab at SickKids. And although you don't see her when your kids go for scanning, most of the time she's the brains behind a lot of these tasks and the analysis that is required for the imaging work that we do. So welcome.
So I'm going to give you a little bit of an update on some of the neuroimaging we're doing. We do a lot of neuroimaging in PON, and so I'm just going to touch on a few things. And some of them you've heard about with the previous speaker, with Danielle, and you'll also hear some more from Dr. Amos, who's speaking right after me. So you all know that MRI provides us with very, very detailed images of the brain. We can get absolutely wonderful resolution of the different structures in the brain. We can go in and measure those structures and do analyses and look at them individually as well as at the group level. And what I'm going to start with is an MRI measures in ASD. We have far more ASD participants. The recruitment for ADHD and OCD has lagged behind a bit. It's better now, but this first study I'm going to present was published a year and a half ago, and so now we have many more data. But this is an important study because there was a lot of initial publications that showed different types of results in ASD in terms of brain structure. And a lot of the studies contradicted each other, and we think this is partly because they had small numbers, and this is one of the powers of the PON group, is that we're getting better numbers and larger samples. And so the one I'm going to show you has 72 children. We now have more than double that number and can do more analyses. So one of the simplest measures we can do, we can take the brain volume, and there is this issue about children with autism having different sizes of brains compared to controls. And in our sample, we don't see evidence of that. You can see here in the, does my mouse show up? No, it doesn't. Okay. On your left, you can see that, so in pink are the controls, in blue are the ASDs. There's a small group effect. It doesn't change with age. The brain gets bigger in the teenagers than in the younger children, but there is not bigger brain in the young children with ASD that has been shown in the literature. When we look on, and you look on the right side, you can see that the white matter, so this is just the white matter in the brain, not the gray matter. There wasn't a difference in terms of the group, but there was a group by sex interaction. And I know Evdokia has mentioned this earlier, that we are going to be investigating some of the sex differences, the differences between boys and girls, because we see this in some of our measures, that difference in white matter was due to that, you can see the lower bar for the girls with autism. So this is a little finding that it wasn't very consistent across all our measures, but we see it in some of these measures. We can also look at cortical thickness. This is the type of image that we get when we're measuring cortical thickness, and this is in collaboration with Dr. Jason Lurch. You can see that we get these very pretty images that measure the differences between groups in terms of how thick that gray matter, the gray matter mantle is. When we analyzed this in terms of the children with and without autism, we saw that, as Danielle mentioned, we get a thinning of the cortex with age. That's normal. Unfortunately, it keeps thinning, and so at my age, it's getting even thinner. But in the young children, it's supposed to thin as the cortex, as the brain gets more specialized. So it's adapting. And you can see that in the two groups, again, the controls are pink and the ASD are blue. We get it going down with age from the preteens to the teens, but there wasn't a significant group effect. But when we went in and looked at more specific areas, and this is a technology that we received guidance in doing with Jason, we can see that in fact in a few key areas in the brain, there is a group by age interaction, where the controls in pink are decreasing their cortical thickness in these two areas. One is the posterior cingulate area and the other is the orbital frontal area. Both these areas are very important in social function, cognitive social function, And the children with autism in blue are not showing the normal thinning. So their brains are staying the same level. They're not evolving in the same way as the controls. And so we don't know the meaning of this, but we're getting indications of changes that are maturational changes that are a little different in this group of children with autism. But we published these data, as I said, almost a year and a half ago, and you can see that we called it diffuse but not pervasive. So we had some effects in some structures and not in other structures, and so it was really nothing that we could say, aha, with that we can diagnose. We cannot do that at all, but we're trying to understand the brain-behavior relations. And the nice thing about the POND data is that we now have large enough numbers to include ADHD, OCD, and more than double the number of ASD to do more comprehensive analyses of the structure. Another measure of structure that we can get from the MRI is the white matter of the brain. And so the gray matter you think of as the thinking material of your brain, your gray matter, but the white matter is the information highways, if you think of a road map in your brain. And we can look at those and measure how efficient they are. It's sort of, if you think of it in terms of the roads of Toronto right now, with all the construction, they're not very efficient compared to a road that's been totally new and working superbly. And what we see in this image here shows you all the areas in green are some of the major fiber tracts, and these major white matter tracts in the brain. And the areas that are marked in red are areas where those major fiber or white matter tracts are working less well in children with autism. So they seem to be less efficient. And when we looked at it in terms of another, you can do many different measures on DTI. And by the way, the next speaker, Dr. Amos, is going to give you much more detailed information about this. She'll describe this in more detail. But you can see that it is quite extensive in terms of the deep white matter in the brain. And it was consistent with fiber coherence, not myelination problems. We also looked at this over age because the question is this just a maturational lag? You know, because what myelin, the white matter, matures right up until the 30s in life. It keeps getting better and better, if you will, up until you're in the 30s. And we did not see a change in terms of the maturational course between the children with autism and the controls. They were parallel to each other, but those with autism were just a bit below, but they were still showing maturation. We now have, with other studies that we're doing with Eptakia and others, a large group of adults with autism as well, and we'll see they may come together. I mean, we have to look at those data. But again, and this is something that Dr. Amos will present, we now have the data in ADHD and OCD as well, so that we can look at cross disorders and the commonalities and differences. So MRI can also be used to investigate brain function. And there's a lot of work on this. And what I'm going to show you is just a single example. I know we don't have a lot of time today. And it's a dynamic faces task. And so this is a task where the children just have to lie in the scanner and they're just looking at faces. And the faces go from a neutral to a smile or from a neutral to an angry, okay? And so there are all kinds of different faces, so it's not like the same face over and over and over again. But they're all they have to do is watch the faces. And we got some very different findings in the 3 different NDDs. You can see on your left that the OCDs have some decrease. In blue, it's a decrease of activation compared to controls for the angry faces. It's in the parietal region. You know, that's a cognitive association area. The ADHD have an increase of activation. So you can see the pattern is very different for the 3, which is the first take-home message. And so that is an area, the anterior cingulate, that is used for monitoring, paying attention. So we think the ADHD had to work hard at paying attention, and that's why they're activating that area more. And the ASD have a decrease in the frontal areas, which is known to be related to social cognitive function. And when we looked at happy faces, the OCD and ADHD had similar patterns. The ADHD are still trying to pay attention. But the ASD, who have the largest deficits in emotional processing, have a very different pattern depending on the emotion that's being presented, suggesting that they are not processing these emotional faces the same way. They're having more difficulty, perhaps, with the different categories of face. And in the happy faces, interestingly enough, that all that orange area that you see in this bottom right here, that's part of the salience network. And the salience network has been involved and been implicated as an area that has is sort of a key area of atypical functioning in children with autism. But these data are still under analysis. This is not the final analysis. We are still working on these data, so we'll have to see what the final outcome is with our results. But it's enthusiastic that we have it across the three disorders, and we can look at different and distinct patterns across them. So I'm going to just focus briefly on MEG. Most of you probably don't know magnetoencephalography. You can see it's a much smaller scanner than an MRI, and it has a number of advantages. It provides very, very good timing information, so we can get when things happen in the brain. fMRI doesn't give us that at all. It just says where things happen. So we can get that little line on the bottom is 1 second. So you can see activation that's greater in one group than the other in about 400 milliseconds. We can see where it's happening, so we can localize when there's that increase, where is it coming from in the brain? That's like MRI. The MEG signals look like EEG, so if those of you who have seen an EEG, you know that it looks very similar, but it is easier to record accurately. And as I mentioned, it's a much smaller scanner. It's totally silent, so it's very easy for all of the children. Those with anxiety in particular find the MEG much easier. And we can use a wide range of paradigms to map cognitive function. All kinds of different tasks can be run easily in MEG. And we can look at connectivity as well. So we can see how the different areas of the brain are connected. And that can be done in fMRI as well. We have examples of that. But the MEG gives us the timing of that connectivity. And of course, in terms of developmental disorders, then you wonder whether there's processing is slower in some circumstances, or it's too rapid in others, depending on the disorder or what you're assessing. And the MEG gives us the power to look at that type of activity. I'm not going to go through much. I just want to mention some of our ongoing studies. We're looking at theory of mind, which is a social cognitive task. We've done it in children. We're doing it in adults. We're looking at an emotional regulation task as well, and doing that across all three disorders. We're looking at mental flexibility, like the ability to shift, to change a way of thinking, which is very important, particularly for the OCD and ASD groups. And we also are looking at interventions, like doing some neuroimaging with various interventions. And I think that this is something that is a big aim in the future, and in terms of where we would go in the future, this would be a huge area where it'd be very important to investigate. I know there have been some questions about this already from you, that the looking at brain function, this is mainly a disorder of brain function. The structure is not blatantly different in these groups, as Evdokia has mentioned, but it's a change in function. And if behavior changes, then brain function changes. And so measures of brain function will be able to monitor the success of the interventions. So these three technologies that I talked about, MEG, MRI, and a little bit about fMRI, are complementary to understand the neural underpinnings, what's going on in the brain to produce the behaviors. We can do quite sophisticated protocols to look at these complex social cognitive abilities. It's starting even in young children. And the atypical brain function can be seen in a range of tasks, and I think bringing these together will help us understand the heterogeneity as well as the overlap in these disorders. And most importantly, looking at the brain function will allow us to assess and monitor both the developmental trajectory when we have the longitudinal data available, as well as interventions. I just want to thank the group. There's a big team that works with neuroimaging. It's complex work to do, and these are the people who have done the work. Thank you.
But it's a really good question, right? So if we're going to pull information across the different groups and the different scientists around the world to create these large databases where we can explore all the different variation, if that's what missing is, then there is an effort to combine imaging information around the world for our kids in a series of initiatives. The most known one is something called Enigma, that is an terrible name for an international initiative where we're all contributing our imaging data so that we can end up with these consortia, that these groups of data, that scans that are account in the thousands and not in the 15, 20, 100 scans, to start answering these questions. Now, it's by no means is as evolved as with missing.
You talk about small groups all the time.
Yes.
But inaccuracies that they've got to be out there
Yeah,
somewhere.
so there are some concerns that have to do, we have to resolve some ethical questions before we are allowed to share scans from site to site. I think it's, so for example, certain ethics board will argue with us that the scan is identifiable information, that somebody, for example, can reconstruct, if they want it and they were malicious, they can reconstruct from all the slices the face of a kid. And so there are some technical work that's being done to allow for much more sharing. So that's large international initiatives. You should know, though, that our de-identified images, every child that has a POND scan, their de-identified image will go into BrainCode, which is the OBI's effort at sharing data to create those large initiatives. So BrainCode is a public domain database that has governance and rules about who can access and how to protect the information. We put our de-identified scans there so other people will be able in a safe manner combine data with our data at some point. So answer to your question is we're trying. We're not as advanced as with genetics in terms of what it would take to have an international pool.
And that is for the MRI data. For MEG, it's much more difficult. It's much more based on the system, and there are far fewer in the world. And so there's very much more limited data. And here with POND, we have the world's largest data set already for in neuroimaging data and MEG in these disorders.
The MRI study that showed the smiling and frowning faces, was that the same study that showed that had pictures of opening flowers?
Yes.
Good. I was in that study.
Thank you very much.
Do you know when or if you will have any results on that?
We had thought we'd have it finished by now, but there was a glitch in our analysis pipeline. And so the data are in a month or two. So it's interesting, this participant at the back mentioned that they're opening and closing flowers, and the reason is that we had dynamic faces, and so there's movement. So we had to have something else that was a biologically relevant movement to control for movement in the stimuli. And so we used flowers that opened as our control condition.
One more.
So you mentioned before, I think it was for the MEG, that it can help to look at how the brain processes information. And I know for my daughter in particular, she has a processing disorder along with some of her other issues that she has. Will this at some point be able to help in the education process for kids?
I think it might. For example, when we were doing the theory of mind task, so theory of mind is the ability to understand that other people have ideas and beliefs different from one's own. Children with ASD in particular are very weak in that domain, but so are other children in other disorders. We're doing preterms as well, and they also experience that difficulty. We found that with the children with autism, what the MEG data showed is that rather than using what we know is the mentalizing network, they were using their working memory network. So they were working hard using working memory to do this task, and so we'd be able to say, okay, to improve that, maybe we can train their mentalizing network, or maybe we just have to help them with their working memory. You know, we get ideas of what other strategies they're using. And so then we would know what strategies they're using, and that may help with people who are skilled at developing interventions. Yeah, so we felt enthusiastic about that.